In the previous video, we discussed how all carbons from the foodstuff get completely oxidized to carbon dioxide. In this video, we are going to discuss how reduced coenzymes NADH and FADH2 will get reoxidized to NAD+ and FAD. Electrons from reduced coenzyme will ultimately reach oxygen, where oxygen will be transformed into molecules of water. We also will discuss how ATP synthesis is powered by a difference in concentration of protons. In previous discussions, we have learned that enzymes will use the hydrolysis of ATP energy to transform reactions that are endergonic, those with positive delta G, into exergonic reactions, those with negative delta G. ATP hydrolysis releases minus 7.3 kilocalories per mole. A similar amount of energy is also released by hydrolysis of a thioester bond. Since the synthesis of ATP and the hydrolysis of ATP are reversible reactions, synthesis of ATP will require an input of energy. Reoxidation of coenzymes and ATP synthesis are coupled by a proton gradient across the inner membrane of the mitochondria. The transfer of electrons from reduced coenzymes to proteins embedded in the inner membrane leads to the pumping of protons from the matrix to the intermembrane space. Hydrolysis of a phosphoanhydride bond releases energy. When protons return back into the matrix, there is a large conformational change in ATP synthase, the enzyme in charge of the formation of a phosphoanhydride bond in the molecule of ATP. The energy of the proton gradient is sufficient. What are the requirements for the synthesis of ATP? The first requirement is a closed compartment. We will have reduced coenzyme providing electrons. Those electrons will go to proteins embedded in the inner membrane. When those electrons are transferred to those proteins, protons will be pumped to the intermembrane space. Those electrons will go ultimately to oxygen, where oxygen will be reduced to molecules of water. At that point, the concentration of protons will be higher in the intermembrane space than in the matrix. Protons will return back into the matrix by a channel in ATP synthase. When protons return back into the matrix, a large conformational change in 3 subunits of this enzyme will produce ATP that can be released. The electron transport chain is a series of proteins embedded in the inner membrane of the mitochondria that allows the passage of electrons coming from reduced coenzyme. Those electrons will pass from one protein to the other until they reach molecules of oxygen, and oxygen gets reduced to molecules of water. The coenzymes themselves will not enter the proteins, only the electrons. Those proteins will contain centers, coenzymes, and cofactors changing in oxidation state. For example, iron 3 plus will accept one electron and can change to iron 2 plus. Copper 2 plus will accept one electron, changing to copper 1 plus. When the protein is charged with one electron, protons will be pumped. In this schematic representation of the mitochondria, we observe the outer membrane. We also see the inner membrane and the proteins that are electron carriers. We call those proteins complexes. Complex number 1, 3, and 4 can carry electrons, and at the same time they can pump protons to the intermembrane space. Complex number 2 is an enzyme that is in charge of the conversion from succinate to fumarate during the citric acid cycle. This one does not pump protons to the intermembrane space. We also have the complex number 4 contains a heme that can bind oxygen where oxygen can be reduced to water. Complex number 5 represents ATP synthase. This is the enzyme that allows the passage of protons back into the matrix and also the synthesized ATP. After electrons are in complex number 1, how do they travel from complex number 1 to complex number 3? The answer for that is the molecule of ubiquinone or coenzyme Q. Electrons will be sent to complex 3 by use of this molecule. These two ketone groups will be reduced to alcohol group, so we will have 2 electrons and 2 protons to the molecule of ubiquinone becoming ubiquinol. This ubiquinol will send electrons, 2 electrons at a time, and then the electrons from complex number 3 need to go to complex number 4 by use of what is called cytochromes. Coenzymes found in complex number 1 well fixed. They are not traveling through the membrane. However, the molecule of coenzyme Q is a mobile electron carrier. It has a long hydrocarbon chain that it can intermingle with the tails of phospholipids forming this membrane. Among the centers that can bind electrons, we will find cytochromes. And cytochromes are proteins, enzymes that will not be catalyzing reactions, but they will be accepting electrons in the electron transport chain. They will contain heme. We have seen heme in the molecule of hemoglobin that was carrying oxygen, but in this case we will have an iron center that will be changing the oxidation state from 3 plus when it accepts an electron becomes iron 2 plus until it gives the electron away. There are different kinds of cytochromes. Cytochromes in complex number 3 will be accepting electrons from ubiquinone. Cytochrome C is a water-soluble cytochrome that is in the intermembrane space. This cytochrome can travel from complex number 3 to complex number 4. One of the cytochromes on complex number 4 can bind oxygen, and that is where oxygen will be reduced to molecules of water. After electrons are delivered by ubiquinone to complex number 3, those electrons need to reach complex number 4. Those electrons will be sent by what is called a mobile cytochrome C. Cytochrome C is a protein water-soluble found in the intermembrane space. In all 3 complexes, protons are being pumped to the intermembrane space. Only complex number 3 and complex number 4 will contain cytochromes. Summarizing the electron transport chain, we can say that reduced coenzyme will send the electrons to proteins embedded. Those electrons will be sent by ubiquinone to complex number 3. In this process, protons are being pumped to the intermembrane space. Cytochrome C will take electrons from complex number 3, where these electrons are going to be sent back to complex number 4, where oxygen can be reduced to molecules of water. And now we have created a proton gradient. It is when those protons return back into the matrix that ATP molecules will be synthesized.